Okay, so this is chapter 6, and it's a tour of the cell, and it's actually just the beginning of the tour. Today I think it's going to be quite general information, but in this course we're going to actually explore cells quite deep, I should say. So first of all, what is a cell? A cell is a basic structural and functional unit of life. Cell is basic. I think I told you that it's enough to be made of one cell to be considered a living organism. So unicellular, which means made of one cell. So unicellular. Unicellular, like bacteria. And multicellular. Does anybody know how many cells actually we have? How many cells make up our organism? No, talk to me, give me a number.
Professor, is it 4?
How many cells make our organism, our body?
Yeah.
No, there are 4 types of tissues that make our body. You close,
Oh.
but in terms of number of cells.
Trillion or something like that, 72.
70, yes, Jamari, 75 trillions. That's how many cells. And you know, so it's really interesting. On our body, we have more cells that don't belong to us. Actually, bacteria that live in our body, on our skin, in our mouth, in our intestines, actually even more than 75 trillions. So that's like kind of mind blowing, but don't worry about this right now. But life starts with one cell. Whether it's going to continue to be unicellular or it's going to become multicellular, so the cell is going to divide and differentiate into different types to form entire organism. One more time, it all starts with one, right? Even like for humans, we start with one cell at the time of conception when genetic material from both parents join to form the first cell. What do you think? How long did you spend being one cell only? I'm sure you don't remember that, for sure, right? But that's the part of our biography, actually, being one cell. What do you think? How long? No?
Is it 5 days? I'm just
It's actually, I should say, 3 hours. Before the first division, before the cell went into the first division and yielded 2 cells and then 4 and then 8 and more. Yeah, spend like 3 hours as one cell only. Okay. And let me go back to PowerPoint. So when you talk about a cell, so this is infamous, it's infamous, forgot what's name. I remember it's in Russian, forgot it's in English. But this is protozoa, okay? Usually you take a look at it under the microscope during the lab sessions. And this is a plant cell, right? And actually this plant cell, I guess diagram is used just to show you the complexity of the cell. We're gonna talk more about part of it, like different parts. The whole study actually of cell biology definitely started with invention of the microscope. Most of the cells are too small to be seen without any help, right? Without any magnification. Let me see. The only cell that you can actually see without the help of microscope is, anybody knows? Something that you're very familiar with, but it's made of only one cell. Look at this. This is a
chicken egg.
chicken egg. Do you know that chicken egg is only one cell? You know that? Yes, it's an egg cell, right? So for example, if you like think this is a frog egg cell, right? So frog egg cells, the frogs lay for, to be fertilized, right? You can also see these eggs without the microscope as well, right? Fish eggs, human egg cell. Human egg cell is the largest cell human body can produce. I mean, good OBGYN can see it on the ultrasound, right? You can identify the egg cell. If you like take a pen and touch the tip of the pen over the paper, that's the size of the human egg cell. As I said, you can actually see it without microscope. Here's the one. Awesome. I like it. I like you doing that. But yes, like birds' eggs, definitely the largest were dinosaurs, right? Which bird actually in these days lays the biggest eggs? Good morning, Gabriel.
Good morning. How are you?
Good. Thank you. Good, right? 100 on the quiz.
Oh yeah, thank you.
Yes, guys. My question to you: was it too much? 40 minutes, or it was right time for you?
It was all right.
It was all right. So should I leave it 40 minutes for the quizzes?
Yeah, 40 minutes. I think it's good.
Oh, okay, awesome. So now, who can tell me which bird nowadays lays the biggest eggs?
Ostrich.
Yes, ostrich and penguins also, they have big eggs, right? So why it is so big? Why birds and reptiles actually lay big eggs?
I mean, I'm assuming, sorry, when the baby is forming, it needs to grow so it has enough space for when it's fully formed. Therefore, it's always
Absolutely.
big as it would be formed. Yeah.
Yes. Like, for mammals, development is internal, right? Inside the mother. So once the placenta is formed, the embryo, the fetus, receive nutrients directly from the mother. When you lay the eggs, right, you kind of leave them on their own for development. So you have to provide with nutrients, with whatever is necessary for development. That's why the eggs are big, right? As I said, different size for frogs, fish, for reptiles, for birds. And how long does it take for a chick to hatch from the egg? Did you ever wonder? It's 3 weeks. Actually, the same as, for example, mouse pregnancy. Like pregnant mouse goes pregnant for 3 weeks. The same goes for chick to hatch from the egg. And my last question about the eggs, I'm gonna stop bothering you. Those eggs that are sold at the grocery store, can you get chicks out of them if you put them in the incubator and keep them warm and happy for 3 weeks? Can you? No,
No.
guys. No, right. Why?
Because they're not fertilized.
Exactly. They are not fertilized, right? So Like mammals ovulate, right? No matter whether they get fertilized or not, so they just ovulate. Birds lay eggs regardless whether this egg is fertilized or not, right? But usually, for consumption, people take non-fertilized eggs rather than fertilized. And this actually shows you like a scale, right? So human height, right? Some cells in human body, like neurons, they are quite, I shouldn't say large, long, right? So neurons, for example, in your spinal cord, they have processes that could be, like 3 feet long. Like the nerve can go, which is a part of the neuron, can go from spinal cord to the heel. So this is a chicken egg. This is a frog egg. Human egg cell, as I said, the largest. Most plant and animal cells, they are within the range from 1 micron to 100 microns, right? Plants, prokaryotic cells like bacteria, they're usually much smaller than eukaryotic cells, much smaller. LM is light microscope, EM is electron microscope. So parts of their cells, viruses, you can see with a regular microscope. You need electron microscope. And molecules and atoms you can't see even with an electron microscope, right? So they are way too small to be visualized. I'm gonna leave this part for your lab. I'm sure you're gonna talk about microscopy, but what you can see here, like certain pictures, there are different ways how to present biological specimens to take a look under the microscope. Different degrees of resolution. For example, this, what you can see is kind of like clear, right? This specimens are stained. Stained means special color, special dyes were added actually to see the parts, to visualize, to make them brighter, right? More clear. This is transmission electron microscopy. This is scanning. This have to be fixed before you actually take a look at them under this type of the microscopes. Fixed means, again, certain chemicals on top of dyes are added. This is a protein, and this is cryo-electron microscopy. Again, another technique to see parts of the cell. And fractionation is actually to separate parts of the cell apart. And centrifugation is a technique actually that separates contents according to their weight. It's called differential centrifugation. Centrifuge, the one that like spins fast. And by spinning, you can actually separate the contents by weight. The heaviest go to the bottom and the lightest go to the top. And this is actually how you can determine the makeup of the cell, the number of the organelles and the function of the organelles. So this is a tissue. You have many cells. You mix them. That's called homogenization. Then you spin them in the centrifuge, and then you get the contents separated, like again, separating cellular parts. Okay, now what I want to talk about, first of all, sorry for jumping. I'm gonna go into the whiteboard, and we're gonna talk about, first of all, 2 types of cells. So 2 types of cells. So 2 major types of cells are prokaryotes. Prokaryotes and eukaryotes. So karyo means nucleus. Okay. Pro means before, U means with. Okay. So prokaryotes means cells that were formed, like existed, before those that have a nucleus, which means no nucleus. Eukaryotes with nucleus. What is a nucleus? If you look at the eukaryotic cell, right, you see normally under the microscope 4 major components. You see membrane, which is the border of the cell, right? You see large piece inside called nucleus. You see the interior of the cell called cytoplasm. You see a bunch of weirdly shaped pieces. They're called organelles. So these are 4 basic parts of eukaryotic cell. Okay. Nucleus is a very large piece, large structure, because inside the nucleus you have genetic material. The more complex organism, the more genetic material is inside the nucleus, right? Again, it's a very, very big part of the cell. Prokaryotes are unicellular. They're made of only one cell. So first of all, size of prokaryotes is much, much smaller. These are much, much tinier cells, okay? You don't need too much genetic material for single cell organism. So DNA, which again is a genetic material, usually is found like floating in the cytoplasm, not surrounded, not protected. So DNA, genetic material, is not organized into the nucleus because there's no need. You know, this cell is tiny, doesn't have that many parts, doesn't require too many genes actually to control it. Okay, so genetic material is not organized into nucleus. So pretty much again, eukaryotes and prokaryotes, the major difference is complexity, right? Even though some eukaryotic cells actually, some single cell organisms also belong to eukaryotes. Still, cells much, much more advanced, okay? So difference, one more time, complexity. Second difference is the way genetic material is organized, okay? Another difference definitely is the size. And if I go back to PowerPoint, so here, classification, the major classification of living organisms, you probably learned. Did you talk about this in lab? Did you? I'm not sure if you did. Actually, you know what? Let me go back. Sorry. Let me go back to whiteboard. Okay. You talk about domains in the lab? Yes? No? No. Okay, so domains, you have bacteria, you have archaea. So these are prokaryotes. And you have eukarya. So these are eukaryotes. And here you have protists, you have fungi, you have plants, and you have animals. Okay. So in this world, prokaryotes include bacteria and archaea. Archaea, organisms that look much like bacteria, but they are different in terms of proteins and genetic material. Eukarya, all eukaryotes, and eukaryotes include protists, which also single-celled organisms. Fungi that could be one cell, could be many cells. Plants and animals that are multicellular. I don't think in Bio 1 in 210 you really talk about classification much. I think this is more like for Bio 2. But I know that sometimes in the lab, one of the labs actually, when you talk about classification as well. Anyways. So let's take a look at the prokaryotic cell one more time. This is bacteria, typical rod-shaped bacteria. Bacteria classified by shape, like round, spirals, rod-like. This is a typical rod-like, I should say. This is a typical E. coli. Have you heard about E. coli? Yeah, the one that lives in large intestine, okay? And if you look at this one, you see that it has plasma membrane, right? But on top of plasma membrane, there's something called cell wall. These are tiny creatures. They need support and protection. So on top of the plasma membrane, they're usually surrounded by cell wall. Quite often you can find something like hairs called fimbria. These are to attach to the surface, like to stick to surface. You can find tails, it's called flagella. And this is a nucleoid. Nucleoid is a region inside the prokaryotic cell where is genetic material found. So it's not nucleus, it's just a location, it's like a spot inside the cell where you can find genes called nucleoid. You can find, this is like a cytoplasm, and in terms of organelles, they're really, really limited. So most of the organelles found in eukaryotic cell, you can really find in prokaryotic, okay? This is actually, I was wrong. This is from the same family, didn't pay attention. This is the one that causes diphtheria. Have you heard about diphtheria?
I've never heard of it.
Diphtheria, okay. It's first of all, it's very, very awesome that you never heard about it. It's a nasty disease. It's normally you get DPT shot. Diphtheria, pertussis, tetanus. This is the shot kids get, okay? Diphtheria is a nasty infection that causes in the throat, they like form like a film, like a layer of cell that blocks respiration. Nowadays, it could be easily treated with antibiotics. In older days, doctors, to save children from diphtheria, they had to suck these films out of their throats. So they use special, like, you know, straws for taking that out. And many doctors actually contracted diphtheria themselves and died because nobody cleared their throats for them. Okay, guys, are you with me? I know you're a very quiet class, but still, yeah? Are you with me?
Yes.
I'm here.
Are you sure?
Okay, so I'm gonna like jump to this one. This is prokaryotic cell. So when you talk, sorry, eukaryotic cell, sorry. So when you talk about eukaryotic cell, you usually talk about animal cell and plant cell. They share a lot of structures together, but plant cell has some extra structures not found in animal cells. So this is the panoramic view, and I'm sure you learned about cell before. I'm sure you heard. Anyone here? I mean, if I ask you, can you raise your hand if you never heard about mitochondria? Raise your hand if you never ever heard in your life. Okay, Ophelia, that's fine. I'm gonna explain what is mitochondria. Anyone here who never heard about
All I know is that
Yes.
is the powerhouse of the cell, the mitochondria.
Awesome, Ken. Awesome. Yes, you're absolutely right. It's the powerhouse of the cell. Anyone here who never heard about chloroplasts? Chloroplasts? Yeah? No? Okay. No problem. Okay, so panoramic view of animal cell. I really like to compare cell to city, okay? Why? Because like city, cell can provide itself with different functions. It's an independent unit, right? So cells can be on their own. Cells can cooperate with each other, but at the same time, cells are quite self-sufficient. So first thing that you see, again, if you look at the cell under the microscope, you see plasma membrane. Plasma membrane, it's a border. It's a border between whatever's outside the cell and whatever's inside the cell. Plasma membrane protects the cell, keeps the cell contents together, and also controls transportation. Whatever enters the cell, whatever leaves the cell. If you compare it to city, I should say in Europe, not really in America, you can find cities, old cities with city walls, right? A long time ago, they were building walls around the city. Like, I'm sure you heard about Moscow, right? It's the capital of Russia. I'm sure you heard about Kremlin. Have you heard about Kremlin? Wait, what's Kremlin? Guys, can you tell me what is Kremlin? Okay, Kremlin is the center of Moscow, where is the Russian government. And Kremlin is actually, it's a city wall, right? So wall built around very, very old Moscow, and it has gates. It has towers, many towers. So gates to enter and exit, towers to watch out, right? To control whoever enters, whoever leaves, right? So like a city wall. Then, as I said, central piece is nucleus. So nucleus contains genetic material, and this is the most precious part of the cell. If you can manipulate genetic material, you can manipulate the whole entire cell, right? So for this, it has a double membrane. Double membrane also to control whatever enters the nucleus, whatever leaves the nucleus. I should compare nucleus to city hall, right? Keeping information about the whole entire city. Around the nucleus, you see organelles. Again, small organs that have different functions. We're gonna talk about them, but they are within like a gooey fluid, which is really much like a white of the egg, right? This fluid is called cytoplasm. Actually, the whole area between the nucleus and the cell membrane is called cytosol. Cytosol is just the area, right? Space. This space is filled with a fluid called cytoplasm. I'm sure you know that, you heard that human body is 50% made of water, yes? Yes, you heard about that?
Yes.
Yes, so most of the water in your body is actually inside your cells. That's where the water is found. 2/3 of water in your body is locked as a part of the cytoplasm, okay? So in the cytoplasm, you find the organelles. You see this big one? huge one, right? It's outside the nucleus, is called endoplasmic reticulum. Horrible name, I know. But the whole space here is for protein synthesis. I think last week I told you that the most important function of the cell is to make proteins. So you can say this is like industrial zone, right? Like a grounds or the factory where merchandise is made, right? And the major merchandise is proteins, right? Smooth, like rough endoplasmic reticulum for protein synthesis. Smooth is for synthesis of other types of molecules. But smooth is much, much smaller because again, most of the space is for protein synthesis. These tiny dots, you can find them on their endoplasmic reticulum. You can also find them floating inside the so-called ribosomes. Ribosomes are actual protein-making machines. So that's why rough, it gives like appearance of sandpaper, all these ribosomes. This structure called Golgi apparatus. It's usually found next to endoplasmic reticulum. Golgi apparatus is to process proteins. Remember, for protein to function, it has to be folded. It has to acquire 3D, right? Tertiary, quaternary shape. So acquiring the shape is right here in the Golgi apparatus. To keep these things going, right, you need energy. And this is mitochondria right here. Cells have to stay clean. Sanitation, you have organelles like lysosomes, peroxisomes. This is lysosome, this is peroxisome, right? So they remove any debris, right, from the cell. Then cells can divide, like grow, divide. You have organelle that is involved in division, that's called centrosome, okay? So, one more time, city wall, city hall, industrial zone, this can compare to, I don't know, again, processing, shipment, energy, sanitation, growth, and expansion, division, right? Pretty much again, as a city.
Professor, what did you say sanitation?
What?
Which one did you say was sanitation?
Lysosomes. Don't worry, we're gonna talk a lot about it. This is like an overview, like a big picture, but we're gonna talk about details in a second. And cells might have like accessory structures, like a tail to move, or like these structures, finger-like projections, microvilli to increase surface area, but we're gonna leave it for now. That's like the introduction. Plasma membrane, nucleus, cytoplasm, organelles. I think I figured out how to share sound so we can actually watch the video together.
Even if you're just studying, a lot of work is going on in your cells.
Can you hear it now? Yes? Okay, awesome.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. When sugars produced by photosynthesis are broken down, their energy is used to make ATP in mitochondria. ATP powers the work of the plant cell. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
Okay, I'm here. And so, as I said, the central part of the cell is the nucleus, right? And genetic material inside the nucleus is stored in form of molecule called DNA. DNA is extremely large molecule, super large. So it has to be packed really, really tightly. So what you can see here is a DNA. So it's wrapped around proteins called histones. So histone proteins are used to pack DNA. And when cell is not dividing, DNA inside the nucleus is called chromatin. Chromatin is, how to say, it's a DNA that you can't really like break apart. When cell is dividing, chromatin is organized into structures called chromosomes. So chromosomes are visible and they are kind of independent units. But when cell is not dividing, you can't see chromosomes. All you can see is tightly packed DNA, which is called chromatin. If you remember from the last class, I hope you do remember, genetic material is all about protein synthesis, right? Do you remember one gene is a code how to make one protein? Yeah, one gene corresponds to one protein, right? So you have information about proteins inside the nucleus, but synthesis of proteins takes place outside the nucleus. As I said, machines that make proteins are called ribosomes. So I think this is here, right? So eukaryotic cell genetic instructions housed in the nucleus and carried out by the ribosomes. So basically information from nucleus has to be delivered to ribosomes, right? So ribosomes can read the instructions and actually build the proteins. And since again, DNA is a super large molecule that can't really be moved outside the nucleus, right? So instructions are copied for each of the proteins. Instructions are copied into a molecule called RNA. RNA can leave the nucleus, get to their ribosomes, either on endoplasmic reticulum or freely floating ribosomes, right? And ribosomes can use the instructions to build a protein. We're going to talk about this in more details later. But what you can see, nucleus has a double membrane. It's called nuclear envelope, which has inner membrane, internal, outer membrane, and the nuclear pores. Pores are openings, and this pore is pretty much for the RNA molecule to move from the nucleus into their, outside to be picked up by the ribosomes, okay? Not too many molecules can actually enter the nucleus. I should say like transportation in and out of the nucleus is more controlled than transportation in and out of the cell. Because one more time, you get access to DNA, you can manipulate genes. Before the ribosomes, what else can we talk? right here you have this like closer look, right? Closer look on the nuclear envelope. So you can see outside, you can see the ribosomes, you can see their nuclear pores, right? You can see these two membranes, and this is something called nuclear lamina. Nuclear lamina, lamina means a layer that lines the nuclear membranes, right? Lines their envelope, and it pretty much helps to maintain the shape of their nucleus, okay? And the matrix is actually like internal frame of the nucleus. There's a nuclear matrix. But again, at this point, remember, chromatin, nuclear envelope, nuclear pores. And what they also see is there's round structure inside the nucleus called nucleolus. Nucleolus is like a small nucleus, and this is the site of synthesis of ribosomal parts, right? Ribosomes, they make proteins, so their parts actually made in the nucleolus. And ribosomes, just remember, these are protein factories. So ribosomes made of proteins, and they also made of something called ribosomal RNA. Let me just actually write a few things for you here so you wouldn't get confused. So DNA, as I said, is a huge molecule, right? And DNA, all genetic material. RNA is copy of one gene, right? So one RNA molecule basically corresponds to information about only one protein, right? One. But there are different types of RNA. While DNA is just a DNA, but there is something called mRNA. mRNA is messenger. Messenger RNA, this is the one that delivers code to ribosomes. Messenger RNA is actually, this is the one that is the copy of one gene. There is a rRNA. R means ribosomal, and this is part of ribosome. And there's also tRNA. This is transfer RNA, and this is the one that say like works with ribosomes as well. Okay, so there's a different types of RNA, and for example, in nucleolus, there's a synthesis of ribosomal RNA, which is the part of the ribosomes. Okay, so ribosomal RNA in protein. Ribosomes have two pieces or two subunits. There's a large subunit, there's a small subunit. This is like computer model, and this is like how ribosomes actually found in the cell, free or on endoplasmic reticulum. These subunits, what they usually do, they float separately. However, when messenger RNA binds to one of the subunits, the second one kind of like jumps on the top, and they initiate protein synthesis. So in eukaryotic cell, there's something called endomembrane synthesis system. Endomembrane system means you have organelles that have their own membranes, like structures that kind of like separate them from the rest of their cytoplasm. So the system basically is made of nuclear envelope, which is a membrane on its own, endoplasmic reticulum, where protein synthesis takes place, Golgi apparatus, lysosomes, vacuoles are structures present in plant cells, and plasma membrane. So what's really special about these structures is that they can produce kind of like, let me actually draw it to you. So if this, for example, let's say plasma membrane, right? So this is the plasma membrane of the cell. It can fold on itself and create a bubble. This bubble called vesicle. Vesicle is, again, it's a tiny bubble made of the same contents as a membrane, right? But it can be loaded with certain things, right? And it's used for transportation. So these vesicles can move around the cell, moving things within the cell, moving things outside the cell. And what I wanted to say, they're like literally like mini vents that could be loaded and could be moved around. If this is a nucleus and this is endoplasmic reticulum can also produce vesicle as well, right? If this is like a Golgi apparatus can also make a vesicle. So this vesicular system is pretty much for transport. You make a bubble, you load it with certain things, and you move it around. Guys, are you with me? Am I too fast?
Yes.
I'm with you, Professor, all the way.
What?
I said I'm with you.
You with me? Am I talking too fast?
No,
No.
Do I make myself clear to you?
so far, yes.
Okay, thank you. You made me feel much better. So another like few minutes and you'll take a break. So you're gonna have a rest from this material. So a little bit more, just a little bit.
Will we finish today?
Yes, we're gonna finish today. Yeah. Okay, so that's about the vesicles. So transportation. Endoplasmic reticulum, I already told you about. This is the structure that surrounds the nucleus, right? Pretty much clings tightly to nuclear envelope. And you have two types. You have rough endoplasmic reticulum. Rough because of the ribosomes that are found there. It looks like a system of tunnels. And by the way, you see this is another vesicle, transport vesicle here. This again, increases surface area hugely, okay? For the sole reason to provide space for protein synthesis. There is a smooth endoplasmic reticulum. Smooth is not for protein synthesis, but for like lipids and steroid synthesis. Yes, it can also do detoxification, drugs, poisons, can store calcium ions in certain cells, okay? But again, think about endoplasmic reticulum as their factory grounds, like where you have machines for synthesis. As I said, rough binds ribosomes and glycoproteins. What you can see here from this name, glyco is usually referred to carbs. Proteins are proteins, and glycoproteins pretty much like keep ribosomes like stuck to like bound to endoplasmic reticulum, rough endoplasmic reticulum. And as I said, it's a factory. Golgi apparatus. Golgi apparatus is usually found next to endoplasmic reticulum. So if this is again synthesis spot, synthesis place, Golgi apparatus, as I said, where protein synthesis is complete. That's where their polypeptides, long molecules, long chains of amino acids, are folded to acquire functional shape. Functional shape, remember, 3D or 4D, tertiary or quaternary shape. So it happens here. Communication between endoplasmic reticulum and Golgi goes through vesicles. The part of the Golgi that faces endoplasmic reticulum is called cis face, facing. That's where they pick up polypeptides. After processing of proteins, they release them inside the vesicles. The shipping side is called transphase. Receiving cis phase, shipping is transphase. The more secretions cells do, the more Golgi apparatus they have. Like, for example, liver cells have a lot of them because liver cells secrete a bunch of things. Cells that produce saliva also have a lot of Golgi apparatus or Golgi complexes. Then, as I said, cells have to provide sanitation. The sanitation organelle is called a lysosome, right? So lysosome is also sac, right? It's a membranous sac that has very powerful enzymes that can break down large molecules. I should say like enzymes kind of similar to Clorox, something like this. Some lysosomes actually loaded with these enzymes directly from Golgi apparatus. And let me just show you here, right? So for example, cell takes in certain molecules and forms a vesicle. This vesicle could be called like a food vacuole, right? Vacuole is also like a sac in this sense. They usually fuse with the lysosomes, and lysosomes provide digestion. This usually for cells like amoeba or some like protozoan that take food this way, right? Our white blood cells are able to literally like eat germs, right? So large white blood cells will engulf the germ, take it in, right? Fuse with the lysosome, and that's how they will be killed, broken down, right? And lysosomes can also use these enzymes to break down like faulty organelles, something that is broken inside the cell. They can take in and process. It's called autophagy. Phago means eating, right? Like self-eating. And vacuoles, again, as I said, is pretty much the same as their vesicles, like sacs that could be loaded with proteins or other molecules. Depending on the cell, food vacuoles, again, phagocytosis, taking in food inside the cell. Some microorganisms like protists have vacuoles to release waste products outside the cell or excess of water. Central vacuoles are usually found in the plant cell. That's how plants store water inside. And this is like plant cell, and you can see this is the central vacuole. Actually, when you squeeze fruits or veggies and you get the juice, that's where you get the juice from. You get it from the central vacuole. And this is their, let's watch this video and you take a break after that. This is the endomembrane system.
Protein processing and certain other metabolic activities occur in the endomembrane system. the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are two types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down.
Professor, I had a question.
Yes, please.
So like, do we have to know the difference between lysosome and peroxisome? And if so, like, what's the difference?
Yes, I think there is it's stated here, but I can tell you right now, lysosomes, they usually like after large structures. Peroxisomes, they are after smaller molecules. Peroxisomes usually for proteins, right? While lysosome can take the whole entire organelle and break it down, okay?
Okay.
Okay, guys, so it's 9:48, so I'll see you at 10. Okay?
Right, sounds good.
Okay, see you in 10 minutes. And back to share screen. Again, the wrong one. So, we talked about endomembrane system, again, are organelles that have their own membrane and those that can produce vesicles or larger scale vacuoles from this membranous systems. So we continue with the organelles, and the next two organelles share certain things in common. Even though mitochondria is present in animal and plant cells, chloroplast present only in plant cells and algae. Anyways, cells that are capable of photosynthesis. So chloroplasts are organelles associated with photosynthesis. Mitochondria is the organelle that is associated with ATP production. Now, what are actually common between these two? What is really common is that both mitochondria, chloroplasts, are similar to bacteria cells, and they have their own DNA. And because they have their own DNA, there's a theory called endosymbiont theory, sometimes endosymbiotic theory. And the theory suggests that at a certain point of evolution, the cell that eventually became a eukaryotic cell acquired mitochondria. Other cells acquired both mitochondria and chloroplasts, and they actually became their own organelles. You know what symbiosis means, guys? What symbiosis means?
Is it when an organism binds to its host in order to survive?
Exactly. Symbiosis is like mutual relationship when there are like mutual benefits, like benefits both sides, right? So in this theory, it benefits the eukaryotic cell, it benefits both mitochondria and chloroplasts. I don't know exactly how mitochondria and chloroplasts benefit from that, but probably they do, right? But eukaryotic cells benefits hugely because actually eukaryotic cell without mitochondria can produce only 5% of energy needed. Mitochondria produces the rest of 95%. Okay, and what about the mitochondria? So mitochondria has its two membranes, has outer and inner membrane, right? And it has a lot of folds called crista, okay? So on this crista, there is a matrix, sorry. Nobody's picking up the phone, okay, sorry. So it has cristas, and basically we're gonna talk a lot about mitochondria later. That's where the cellular respiration takes place. Okay, that's where, you can say that's where your oxygen goes to. So your oxygen goes to mitochondria, and that's what's ATP synthesized. Actually, cells that have, you know, how to say, the higher metabolic rate of the cell, the more mitochondria present in the cell. And I don't know if you heard about that, but anybody here who works out or used to work out in the gym? Yeah, Leonard, yeah. Have you heard that if you work out on a regular basis, you burn more calories in between workouts?
I've heard if you say you do a lot of resistance training, afterwards you're still burning like fat after the workout because your muscles are trying to your muscles are broken down and they're trying to repair itself, I think.
That's how your muscles change due to
Right.
workout. But have you heard that you can burn more calories at rest, actually, than compared to when you don't actually exercise?
No.
Okay, so guys, if you think about this, it's actually true. This is absolutely true. Because when you do, especially again, resistance training, right? Your muscles work a lot. What happens to your muscle cells? They produce more mitochondria, right? The more mitochondria they produce, the more calories they're able to burn, right? So even again, let's say you do things that is not related to workout, just walking, actually having more mitochondria makes you burn more calories. This is actually one of the benefits, huge benefits of exercise, okay? So you can actually control, not control, but you can actually change amount of mitochondria in your cells. And let's watch a video. I'm gonna tell you also some interesting stuff about mitochondria as well. Oops, sorry.
All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made.
Okay, so as I said, mitochondria is producing like 95% of all ATP. And this organelle has its own genetic material, has its own DNA. So we have DNA in the nucleus, right? It's called mDNA, nuclear DNA. We have mDNA, mitochondrial DNA. Now, it's really special about mitochondrial DNA is that you inherit it from mother. why do you inherit it from mother? Because egg cell, right? Remember, egg cells are huge. They're huge because they have a lot of stuff. Now, maternal egg cell carries all organelles. So what we're talking about, basically, the lysosomes, the endoplasmic reticulum, their Golgi apparatus, and mitochondria, they all come from the mother, right? Father gives only genetic material, right? Father gives only 23 chromosomes, talking about human. Which means that you get mitochondria from the mother, which means you get DNA from mother, right? So mother actually gives more DNA than father. You follow? So there is a genetic disease that is associated with mutation in mitochondrial DNA. In this case, baby born with this disease gonna have really hard time producing energy. So won't be enough energy to sustain life activities. And this is, again, devastating genetic disease that kids suffer. And for a very long time, there was no treatment. And there's no treatment, actually, I think, for a child that is born like that. But in these days, mother who carries this mitochondrial mutation can have a healthy child. How? Have you heard about babies born to 3 parents? Have you heard about that?
No, I've never heard of that.
Okay, so don't think it's like something like a threesome. No, nothing like that. But
Is it like a surrogate?
okay, so look, this is a picture how the embryos are made. So this is mother egg cell, right? This is father's sperm cell. Now, mother has, again, faulty mitochondria. So what they do, they take a donor, right? A donor egg cell, take out donor's genetic material, okay? Transfer genetic material from the nucleus from mother and father. And here you go. Okay, so basically what they do, they take the donor egg cell with mitochondria and mitochondrial DNA and use parents' genes to insert into the cell, right? So by definition, biological definition of a parent is the one who contributes genetic material, right? So since you contribute DNA, even if it's a mitochondrial DNA, you consider it a parent. So you can see, I think there's yes, there's a pictures. Probably there's a better explanation how it's done. Let me just see. But again, the idea, I think this is a better one. Making a 3-parent embryo. I think this is better, right? So this is their parent, right? With a faulty mitochondrial DNA. So from her egg cell, they take out the nucleus, right? Then parent who contributes mitochondrial DNA. So they remove her nucleus, insert a nucleus from parent, another parent, right? So you have actually egg cell, which is a combination of two mothers, right? And then this egg cell will be fertilized by father's genetic material. Okay, is it clear, guys? How actually how they make babies from 3 parents? Guys?
Yes.
So, yes.
Okay, yeah.
Yeah, I mean, actually, I don't know if you heard that mothers who, like the older the mother, right, the more chances that there will be some genetic problems with the child, right? That maternal age is actually associated with genetic problems. Have you heard about that?
Yes.
Yes,
Yes.
actually, what really increases chances with mitochondrial DNA. Mitochondrial DNA is less stable than nuclear DNA, and problems associated with these genes are more common when mother gets older. So, mitochondria, one more time, power plant of the cell, has its own genetic material. Another organelle with its own genetic material, but present only in plants, chloroplasts. So, photosynthesis. And chloroplasts, like I'm gonna show you like diagram and then I'm gonna watch the video. So in the chloroplast, first of all, what's really like apparent thing, they're green. We're gonna talk about the photosynthesis and you're gonna learn about the green pigments, the chlorophyll, which is present in the chloroplasts. And chloroplasts give the green color to the plants, right? So it also has inner and outer membrane. It has its own ribosomes, and it has something called thylakoids. So thylakoids are structures that literally like a bunch of coins stuck on each other. Space between them is called stroma. And in a thylakoid, actually, you have all the equipment for the photosynthesis right there, okay? Chloroplasts are also called plastids. Plastids are organelles that are present in the plant. So let's watch a video about them.
Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. When sugars produced by photosynthesis are broken down, their energy is used to make ATP in mitochondria. ATP powers the work of the plant cell. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
True, right? The only thing again, animal cells don't have chloroplasts, but plant cells have both mitochondria and chloroplasts. And peroxisomes. So peroxisomes are organelles that actually similar in certain ways to lysosomes, right? They also have enzymes, but they not do the oxidation reactions like lysosomes. They don't have that powerful enzymes to break large structures down. But what they do, they can remove hydrogen atoms and they produce hydrogen peroxide. You know what's hydrogen peroxide? Guys? You know what's hydrogen peroxide?
Yes.
Yeah. What hydrogen peroxide does?
Doesn't it like remove dirt? It produces. It's not oxygen. I don't know how to explain it. I'm sorry.
Yes. Look, what's really interesting about hydrogen peroxide that it's very similar to water molecule, right? So water is H2O, right? Hydrogen peroxide is H2O2, but it's highly reactive, very reactive, very much, so much more than water. So, and first of all, it's a good like disinfectant, right? So if you have a cut, you can apply rubbing alcohol, but it's going to burn. Hydrogen peroxide doesn't burn that much, right? And it also helps to stop bleeding, much better than alcohol, by the way. And it's also bleach, okay? It's a powerful bleach. Your cells produce a bunch of hydrogen peroxide, and especially, for example, cells on your skin. And also cells, you know, the skin cells on your head, which sometimes you get gray hair, and you get gray hair pretty much due to hydrogen peroxide activity because it's a bleach, okay? And so they use, again, this oxygenation reactions, the peroxisons. And this oxidation is actually, as I said, can modify certain chemicals, can break them, molecules can break them down, right? And what do we have here? Yes. So again, they do sanitation, but on a smaller scale than lysosomes. And another part here is cytoskeleton. So what is cytoskeleton? It's internal frame of the cell, right? Like we have skeleton made of our bones to build the rest of the body around. So cells also have their own skeleton, which is made of protein molecules, different degrees of strength, different degree of size, right? But they form internal network inside the cell. So let's take a look.
Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another.
Yes, so again, they provide internal frame. And for example, this larger beams, like beams, they can also be used as highways inside the cell for the vesicles to move around. Okay, and it's not only about support, cytoskeleton also can provide motility. Motility is movement. Okay, so there are parts of the cell that are able to move, like a tail or some smaller structures attached to the cell, and also again, internal movement as well. So this is like, for example, motor protein, and remember I told you vesicles move around. So vesicles don't really glide on top of this parts of the cytoskeleton. They have motor proteins attached. This motor proteins powered by ATP molecules, and you see like tiny legs. Isn't it like really amazing? And guys, keep in mind, that's what happens actually in your cells. There is something that, to tell you the truth, I don't know why, from my experience, like teaching for a long time, students somehow relate better to certain information than to other. Like students usually relate well when you talk about molecules, right? Carbs, proteins. But when you get to cell, it's getting harder. To my opinion, cell material is a breaking point for many bio classes. Some students, once they learn about the cell, they say, okay, this is not for me. You know what, biology is something different from what I thought about. So I don't want to deal with this anymore, right? But again, the best way to acquire information, to learn this information, is to make it relevant to yourself, okay? So when you see all these structures, when you see all these names, try to think that that's what actually, that's your cells in your body, right? This is your muscles, this is your skin cells, this is your neurons, this cells in your heart, okay? What do you think about it, guys? How do you find this material? Can I get some feedback from you, please?
For me, it's a familiar topic, so it's not foreign. So I at least have some background knowledge about it. So I'm catching on really quickly. I'm pretty sure like you're gonna make the test more difficult than like a high school test, but for right now, I think the information is pretty achievable, I want to say.
Okay, thank you, Jamaris. Can I hear another opinion, please? No, talk to me, guys. D1, what do you think?
I don't think it's too bad. I feel like I'll just have to go over a couple of terms and like look it over, but it is like Jamara said, it's not too bad. Like if you look it over, it's something similar to high school. So yeah,
Okay.
but with more information, obviously, you know, more in depth. But yeah.
Yeah. Okay, so not bad. I feel better about this because, as I said, maybe terminology or I remember I had like I was teaching class like a while ago, and then girl came to me after the class and she said, "Do you really mean all this like mitochondria in my cells produce ATP right now? Right now when I'm talking to you?" And I said, "Yes." And she's like, "Oh." So like I don't know. It's probably again something that you never thought about. Like while you sitting, walking, eating, your cells do their work that you have no clue like how they do it, how they know how to do it, right? And what kind of things they're capable of doing. That's sometimes really mind-blowing. Okay, let me go back. So again, vesicles move along the microtubules of the cytoskeleton. And these are called motor proteins that's responsible for transportation. And what are found in the what actually makes up cytoskeleton, what types of fibers. So these are fiber proteins. Fiber proteins means proteins that are like strings. So there are microtubules, which are the thickest, and they, again, make like the internal frame. There are microfilaments, which are also called actin filaments. These are the thinnest components, and they're flexible. Intermediate filaments, again, filaments, the same word as tiny fibers. They have a middle-sized diameter, and they usually found close to the cell membrane. They help to maintain the outer shape of the cell. And cells vary in shapes. They vary in shapes like, for example, neurons that look like stars, skin cells that can look like flat tiles. So let's take a look. Microtubules, right? These are the biggest one, and they provide the shape of the cell, move organelles around the cytoskeleton, and they also involved in cell division, something we're going to talk about later. During cell division, they can separate the cell contents. So these are the biggest microtubules. Then you have microfilaments. As I said, they actually provide flexibility. And intermediate, usually they found close to their membrane and help to anchor certain structures. Also, they can anchor nucleus in the central part of the cell as well. And centrosomes and centrioles. So this is the organelle that can produce microtubules during cell division. So centrosome, this is the name of the organelle that is made of two pieces they call centrioles. So centrioles, they usually found like, quietly inside the cell until cell goes into division, right? And during division, this microtubule is kind of like sprout out of the centrioles and produce something called division spindle. When we're going to talk about the mitosis, that you're going to see the whole spindle actually to arrange and separate chromosomes during the whole process. And structures that basically, you know what, guys, really what's interesting, sorry, Emil, like for me for going over this. Yeah, we're gonna pretty much, you know what, we're almost done. literally, we're almost done with the plant cell. So these are structures that could be associated with their animal cells. So almost done with animal cell, and then I'm gonna talk about plant cell. So what is cilia and flagella? So cilia and flagella are structures that are associated with the cell that can actually capable of movement. Okay. Flagella is literally tail, right? And tail that in human cells, only sperm cells have it for swimming. Cilia are like hair, literally like hair, that's found on the outer membrane of the cell, and they can do this movement, like beating movement, like this. Flagella, when they swim, they propel themselves. Cilia don't move the cell. Cilia move things like certain things on the cell surface. Okay. What is this? Let's take a look at this.
Chlamydomonas is a unicellular green alga with 2 interior flagella and a single cup-shaped chloroplast. Each cell is about 10 micrometers across. This cell is viewed with Nomarski optics, which gives an image that appears three-dimensional. The smooth space at the anterior end, at the bottom of this cell, is the contractile vacuole, which you can see contract in this clip. This organelle is involved in water regulation in Chlamydomonas. By expelling a solution hypotonic to that in the cell, the contractile vacuole counters the osmotic movement of water into the cell. The slightly larger circular structure within the chloroplast at the other end of the cell is the pyrenoid, where starch is deposited. The 3D appearance around the pyrenoid is due to the view of the thylakoid membrane stacks in the chloroplast. Credit, Michael Clayton, University of Wisconsin.
But yes, so this is flagellum beating.
See the complex motion of the flagellum of an immobilized mouse sperm. These images were taken using dark field microscopy, with the recorded image inverted to show the cell as dark against a light background. As the flagellum undulates, notice the presence of smaller secondary waves superimposed upon the large back and forth primary waves. Credit, Geraint Vernon and David Woolley.
Okay, this is a cilia that can help move the paramecium.
Paramecium moves by coordinated beating of its many short cilia. The cilia form longitudinal rows along the entire body of the cell and into the oral groove. The beating action of the cilia moves the paramecium forward and backward. It also produces a slow rotation of the cell as it moves through the water. The cilia in the oral groove propel food particles into the cell mouth, where the food can be ingested by phagocytosis. Credit, Graham R. Kent and Rebecca L. Turner.
Okay, so the thing is that a lot of single cell organisms, they have flagella, again, to propel themselves. They also have cilia for the movement. In mammals, as I said, flagellum is found only in the sperm cells, and cilia moves molecules, substances on top of the cellular surface, okay? What they have in common, right, is that the microtubule actually arranged at the structures in both flagella and cilium, and there is a basal body that anchors both of them, and there is a protein called dynein that provides the movement. Okay, so this is, for example, for, I guess this is for cilia, right? So this is the basal body, right? This is the microtubules. And this is the motor protein called dynein. This is the way it arranged, right? That can provide either like propelling function or actually like sweeping motion. So let's take a look.
Flagella, singular flagellum, and cilia, singular cilium, are microtubular structures that extend outside some cells and that assist in moving the cell or the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is large, whereas a cell will have few flagella or a single flagellum. Many protists have cilia, and the sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulin subunits organized to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP.
Okay. So what I also want to tell you, actually, this is also about movement. So one more thing here is this flagella is clear, right? So flagella is to propel, like this, for example, bacteria cell may have 1, might have 2, might have many, many flagella for movement. What about cilia? Again, relevant to us. Let's say this is your, let's say this is your airways, right? So your airways, if you take a closer look, your airways, they're lined with cells, right? And these cells have cilia. When you inhale the air, right, the air is far from being clear, from being clean. What do you usually inhale with the air?
Oxygen?
Oxygen.
I'm talking, yes, you inhale oxygen, but I'm talking about, like I said, air is not clean. What you can find in the air?
Pollution.
Pollution, yes, dust. What else?
Skin cells.
Yeah, you can find pollen, right?
That's it. You
can find spores of molds, right? Yes. So if all this garbage will go directly to your lungs, right? Your lungs will be useless in no time. They will be just destroyed, right? So when you inhale air, let's say this is the direction of the air. These cells, they have like a sticky fluid on the surface. They trap all this garbage. And cilia actually beats opposite direction, right? So air goes down, but cilia propels everything up, 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 up. So when you do this from time to time, right? What actually happens, whatever was trapped on the way down ends up in your throat. And then it's up to you what you do. What are you gonna do? Yes.
Okay.
So you yes, Jamarius. What are you gonna do?
Throw up.
You can speed it up, right? But I mean, especially if you have a cold, right, and a lot of mucus, a lot of stuff accumulated, right? But usually you just swallow it. No big deal. Your stomach can take care of the things. By the way, a lot of infections people get this way. Airborne infection, they inhale it and then they swallow it. For example, that's how people get chickenpox. No, I mean, like, not in these days because people get shot for chickenpox, but some like viruses, they work that way. What did you want to say? Jamarius, what did you want to say?
Oh, nothing. I didn't say anything.
No? Okay. Now, smoking, nicotine paralyzes the cilia movement. So what happens to the lungs of people who smoke?
They get weaker and kind of deteriorate in a way.
Yes, they start to accumulate all this garbage, and that's what can trigger lung cancer, right? And other diseases associated with lungs. Because, as I said, it's not nicotine itself that is so toxic, but the fact that cilia is paralyzed, right, and can't filter properly the air that we breathe. Okay. And this is microfilaments. So microfilaments or actin. Actin is actually what's called a motor protein. Protein that can contract, right? Get shorter and longer. And they support the cell shape. And as I said, they provide flexibility. And this is microfilaments, actin filaments. These are intermediate. And those that function cell motility, they also have protein called myosin. Actin and myosin in our skeletal, in our muscles, actually responsible for the muscle movement, right? But you have cells that move on their own without flagella and cilia. For example, a cell called amoeba. See, this is amoeba. I'm sure you heard about amoeba at least once in your lifetime. Yes, am I right? You heard about amoeba?
Yes.
Yes, it's a cell that can crawl, right? And it can produce, extend like parts of the cytoplasm. It looks like a leg. It's called pseudopodium. Podium means leg, pseudo means fake, right? And this movement is pretty much due to contraction of proteins called actin and myosin that provide the movement for amoeba. They also provide, again, movement for our cells, our muscles as well. And let's watch this one, cytoplasmic streaming.
In these cells of Elodea, a water plant, the central space of each cell is occupied by a huge central vacuole, around which you can see cytoplasmic streaming. In the stream of cytosol, you can see mitochondria and also chloroplasts, the larger pigmented organelles. This movement allows nutrients and metabolic products to reach all parts of the plant cell. Credit Michael Clayton, University of Wisconsin.
Okay, and intermediate filaments, I already told you, pretty much like able to anchor certain structures on the cell membrane. And what else? We're getting to the plant. So organelles that present in plant but not present in animal cells are, first of all, cell wall. Also remember, cell wall is present in bacteria cells as well, but it's actually different in terms of their molecular makeup, right? So as I said, prokaryotes, fungi, some protists also have cell walls, but it's different from plant cell wall. And the plant cell wall is made of cellulose. We talked about cellulose and molecules like polysaccharides and proteins. So cell wall that looks like this, right, has actually 3 layers. There's a primary cell wall, which is thin, flexible. Then middle lamella, lamella means layer, and it's between the primary walls and has polysaccharides called pectins. And there's a secondary cell wall, not in every single cell, but it's actually between plasma membrane and primary cell wall. So let's take a look. This is the primary, the outer, right? Then there is a secondary, and in between, there's also middle lamina, right? Middle lamina, primary, and secondary cell walls. And in some cells, this actually could be in animal cells. Let me just see. You can find something called extracellular matrix. Extracellular matrix is material that produced by cells, especially like in certain tissues we call connective tissue. In connective tissue, you have cells and you have extracellular matrix. Your bones are made mostly by extracellular matrix that has collagen and calcium salts, right? So you can see collagen, proteoglycans, and fibronectin. These are complex molecules. These are polymers that actually can perform different functions, from storage to structural support. So you can see this is cytoplasm, this is extracellular fluid, and this is extracellular matrix, which could be made by proteins, by carbohydrates, like that. And cell junctions. So cell junctions actually are bind cells together and provide communication or structural support. Plasmadermata are tiny channels that found between plant cells and allow actually water and other fluids and some molecules as well to move from cell to cell. So you can see these two cell walls, right? And these are channels, the plasmadermata between the cells. And other connections which found in animal cells called tight junctions, desmosomes, and gap junctions. So tight junctions, really tight. So you can see two cells and there's really limited space between them. So this is tight junctions. Desmosomes, they are more like anchoring, like binding cell together. And these cells usually found between the neighboring cells, these connections, like tight junctions and desmosomes. Sometimes between the cells, you can find gap junctions. These are gap junctions, tiny channels that allow molecules to move directly from one cell to another. Okay, so this is for multicellular organisms, which means again, cells cooperate, work together, not for single cell for sure. So limiting space between the cells, tight junctions, binding cells together, desmosomes, providing flow of molecules from one cell to another, gap junctions. And this is it. Oh my gosh. So let's just watch the last video and you're almost done.
Animal cells in multicellular tissues are usually joined by tight junctions, desmosomes, also called anchoring junctions, and gap junctions, also called communicating junctions. Plant cells are often joined by plasmodesmata. Each type of junction has specialized functions, such as preventing fluid leakage between membranes or allowing the free passage of small molecules and ions between adjacent cells. Tight junctions, which bind cells together, are found predominantly in epithelial tissues, such as the lining of the intestines. Tight junctions form a barrier that prevents fluids from moving between the intestinal space and the space between cells. Desmosomes, or anchoring junctions, appear as thickened patches in the cell membrane region between two cells. Desmosomes contain specialized proteins, such as keratin, the same protein found in fingernails and hair, that increase the rigidity of tissues. Desmosomes, such as those found in epithelium, smooth muscle, and many other animal tissues, are button-like junctions that bind cells together and also function as anchors for fibers in the cytoskeleton. In gap junctions, or communicating junctions, two cells are separated by a small gap, which is bridged by specialized channels that allow the passage of water and small molecules. Gap junctions help coordinate the activities of adjacent cells. For example, a hormone that stimulates one cell will often activate adjacent cells as well by the passage of intracellular signals through gap junctions. Plant cells, unlike animal cells, are surrounded by thick walls that form rigid tissues. They do not need the junctions found in animal tissues. But some higher plant cells are interconnected by plasmodesmata, tube-like structures that penetrate through cell walls and form fluid connections between adjacent cells. Like gap junctions in animal cells, plasmodesmata allow the free exchange of small molecules and help coordinate the activities of neighboring cells.
Okay, so I think we are, yes, these are just images, okay. So we done. Let me just, the last thing that I want to write down for you is, so plant cells. So what plant cells have that animal cells don't? So plant cells have cell wall, they have central vacuole, they have chloroplasts, and they have plasmodesmata. That's what is not found in animal cells. Okay, so this is the cell, right? So this is the cell wall. Big sac is a central vacuole. Green organelles are chloroplasts. In between adjacent cells, you can find plasmodesmata. Okay, so this is a summary. And any questions, guys?
No, Professor.
No questions. Okay, so last thing that you're gonna do is,
Wait, I'm guessing the test is, I mean, not the test, the quiz is,
chapter 6.
yeah, okay. Just chapter 6, right?
Yes, will be chapter 6. Okay, so.
Are we gonna have a midterm?
Yep, you're gonna have a midterm as well. So let me share. And guys, I want to hear from you because otherwise I can't see the screen. Okay. So Eamon, present here?
Yeah, I'm here.
Okay. Mady, I think you're here, right? Mady?
Yeah, I'm here.
Roshana, I think you're here. Right? Roshana?
I'm here.
Yes. Ija?
Here.
Neela?
I'm here.
Here. Natis is here. Johnny, Elliot? Here?
Here.
Okay. Kevin, I see you. Juliana? Here?
Here.
Okay. Brianna? Here? I think she mailed me. Shalom is here. I saw Jamarius is here. Soraya?
I'm here.
Okay. Ophelia is here. Dulce? Marines? Dulce? Okay. Gabriel is here. William? William Mason? Okay. Azra?
Here.
Here. Olivia is here. Macarena?
I'm here.
Okay. Irandi?
I'm here.
Tiffany? Here, I think. Fehmina? D1 is here. Dominic is here. Leonard is here. And Ebony is here. Okay, guys. So next week, pretty much the same setting. 40 minutes for the quiz on chapter 6, today's chapter. And we're gonna move on. I think we're gonna spend like a while on the cell. Then will be the midterm. I'm gonna announce like at least 2 weeks before the midterm. And Bio 210 is really centered around their like biology of cell. So we're gonna spend time talking about respiration. We're gonna talk about photosynthesis. It's not really much into plants and animals like Bio 220. 220, that's you're gonna talk about animal world. This is like a core biology, pretty much on the cellular level. Okay, so you enjoy